let's start out with, we're going to talk about the nervous system. And as we go through the nervous system, this is the system that is going to be the reason why you are able to do the things that you do. So it's very complicated and very complex, but it's the reason why you are going to eat pizza if you like pizza, why you will remove the pizza from your mouth very quickly if you burn your mouth because the pizza's too hot. A lot of the things that you do every single day are all related to the nervous system, and you just don't know it. So as we go through and talk about how the nervous system works, you're going to get a better understanding of how all of this information is going within your entire body. So if you write down, we always have to start with, first and foremost, some sort of detection. So there has to be some sort of sensory information that is detected. Like if you're hungry for the pizza, the sensation is hunger, okay? Some sort of sensory information has to start this process. And if we then go into the next part, it is some sort of integration. So if you are hungry, that's the sensation. Well, then the next step is, well, what am I going to do with this information? Okay, so that's integration. And then the third step is going to be, okay, well, I'm hungry, sensation. Integration, my brain is telling me that I should go and order some pizza. So the third step is going to be that motor action, the actual act of ordering the pizza. You have to pick up the phone, you have to dial the number, you have to explain with your voice. All of this is nervous system. And that third and final part is that action. Okay, so that's going to be a motor thing that we talk about later on. So it starts with sensory, we have some sort of detection, we then go into integration, and then we go into motor. Okay, those three things are always going to be occurring. That's going to be the reason why we either eat the pizza, why we walk in the rain with an umbrella, or why we snuggle up with a warm blanket whenever it's 20 degrees outside. All of that is nervous system. So for the nervous system, this is going to include the brain and the spinal cord. Of course, those are going to be two very important structures that we're going to be discussing through this module. The receptors. The receptors are going to be in various locations. So we have receptors in our skin that we've kind of already seen through the integumentary system. Those receptors can tell us if something is hot or if something is cold, if something is putting pressure on a certain part of your skin, or if you even have a bug that lands on your skin. So we have different sensory receptors in different places. Eyes, ears, skin, nose, etc. And then nerves that connect to other systems. So these are going to be things that you can find in different places outside of what's already been mentioned. So for example, like in the gut, in the GI tract, there's going to be nerves that are going to connect within the gut to the nervous system. Okay, so we have two kinds of cells. In order for this all to work appropriately, we've got to have neurons that are going to be the big ones that are going to be doing the communicating. The neurons, think of them as the CEOs of the system, okay? The CEOs, there's not that many of them. They have a big job to do. They have major companies to run. So very, very important that we have CEOs who know what they are doing and who do it well in order to keep that business alive. The neuroglia, also known as glial cells, these are going to be like your secretaries. Okay, so we have a CEO and we have a secretary. The neuroglia are going to be absolutely essential to the functioning of the neurons. So basically, they're going to provide the support that the neurons need, whether that's nutrition, whether that's cleanup, whatever those neurons need, the neuroglia are going to be there to do. So we have generally, most textbooks say a 10-to-1 ratio, 10 neuroglia for every 1 neuron. So remember, you have way more secretaries than you have CEOs. Okay, we'll see the different types of neurons. We're also going to see the different types of neuroglia and where those are going to be located as we go through this first portion. There are different divisions, so please make sure you absolutely understand this. The central nervous system, I want you to think about what's in the center of the body. In the center of the body, you have the brain and you have the spinal cord running right down the center of your body. That is the central nervous system. The peripheral nervous system is everything outside of that. So whatever is going to go all the way to the periphery, like to the periphery of your fingers, all the way to your toes, that's going to be peripheral nervous system. Anything outside of the brain and the spinal cord. When we're looking at the central nervous system, of course, we already said brain and spinal cord, so big star, make sure that you remember that. That's what's in the center of the body. The central nervous system is going to have the nervous tissue as well as connective tissue and blood vessels. And its function, big star, is to process and coordinate that sensory data from inside and outside the body. That's step number 1 that we just talked about, that detection of that sensation. So we've got to have that detection that is going to be processed, which was step number 2. The processing is actually occurring in the central nervous system. So the step number 2 is happening inside the CNS, okay? Motor commands control activities of peripheral organs, so sending that motor information out to that hand so that way it can make the phone call. That is going to be motor action, step number 3, okay? Higher functions of the brain include intelligence, memory, learning, and emotion. So anything that's going to be going on within the brain and the spinal cord is going to be technically qualified under the central nervous system. Whereas the peripheral nervous system, again, all the tissue outside of the CNS, and this is ENS, this stands for Enteric, E-N-T-E-R-I-C. E-N-T-E-R-I-C, and enteric nervous system, that is what's in your gut. Okay, so the PNS is everything that's outside of the CNS and the ENS. The purpose of this system is to deliver that sensory information to the CNS. So this was step number 1 that we had in our example. So the delivery of that information is going to occur from the periphery, and that information will be brought to the center. So step number 1 is here in the peripheral. Number 2 is in the central. Okay, and then number 3, this is going to be carrying out that motor command. So these nerves, these peripheral nerves, are going to carry that information out so that you can do whatever it is that the brain and or the spinal cord said needs to happen for that detection that you were made aware of. In our example that we used, the 1, 2, 3, you were aware of something. This is still going to happen though. That whole 1, 2, 3 step process is still going to happen even if you're not aware of it. So we'll talk in a little bit about the things that are going to happen automatically. You'll see how that is going to work even though you don't know that it's always working. So when we're looking at our cranial nerves, these are the nerves that are connected to the brain. Obviously, cranial means brain. Spinal nerves attached to the spinal cord, because again, spinal cord. Okay, so we'll have cranial nerves and spinal nerves that are going to be a part of this whole nervous system movement in order to get us our number 1, number 2, and number 3. We have different divisions of the peripheral nervous system. So the afferent division is what carries the sensory information. Make sure that you circle the A for afferent is sensory. Okay, the efferent, it's often pronounced efferent, but I'm going to say efferent so that way it's very clear to you what I'm saying. Efferent is going to carry motor. You cannot get these two confused, because if you do, you'll be like so lost on test questions, lab practical questions. It will be very hard for you if you mess these two up. So what I would recommend you do is write down on your piece of paper the word same, S-A-M-E, E, and put a little space in between the A and the M. So you have SA, a little bit of space, ME. And the reason why you want to do that is because if you ever get lost or confused, you just remember sensory is afferent. That's the SA. Sensory is afferent. Motor is efferent. That's the ME. So just think of the word same. SAME, sensory, afferent, motor, efferent. So that way you can kind of keep those two separate. That does not mean they do the same thing, because that's actually not what they do. Not functionally, they're going to do two different things, okay? So just using that word just to remember what goes to what. So the afferent division is going to carry that sensory information from the receptors in the peripheral tissues into the central nervous system. Okay, so it's carrying it in. If I feel something on my fingertips, that information is going to travel from the tip of my finger, from the sensory receptors in my skin, all the way to my central nervous system, which is what two organs?
Spine and the brain. Perfect,
spine and brain. All right, now once we're there, we have to do step number 2. What's step number 2? You guys remember?
Integrate.
Integrate, perfect. Okay, so once we're there in the spine and the brain, we have to think, what am I going to do with this information? There's something that's on her fingers. What do we need to do? The efferent division is going to take the motor command from the spine and or the brain and send it back out to either the muscle, glands, or adipose tissue. Let's say on my fingers, the issue is that I felt a spider walk across my finger. The muscles in my hand are going to contract as I try to move my finger and maybe even try to flick the spider off, because that was the sensory information received. If the sensory information received was that I just touched something that was super hot, then I'm going to contract also to move my hand away from something super hot. What if the sensory information was that it's just really hot outside and the temperature has just skyrocketed? Well, now that sensory information, the brain is going to say, well, she doesn't necessarily need to contract her hand to pull away from something, but let's maybe release some water from those glands so that way she can cool her skin. So we have that sweat gland that's going to become active in order to help me cool off, all to maintain what? What's the balance?
Homeostasis.
Homeostasis, perfect, okay? So that's why you want to keep afferent and efferent separate. You understand it. Afferent, we're bringing sensory information into the brain and spinal cord. Efferent, we're telling what is the motor command that needs to happen based upon the information that was received. All right.
Question real quick.
Yes.
You said it's going to adipose tissue as well. Why?
Okay, very good question. We'll pause on that and come back to that. Okay. So efferent divisions include somatic and autonomic. When you think somatic, underline the S, because this is going to control skeletal muscle contractions. That's what I was just talking about with the flick of the finger or the movement away from the hot burn. So the somatic nervous system, always think skeletal, okay? When it comes to autonomic, that word sounds like automatic. So the automatic means these are the things you don't necessarily think about. So this is going to control those subconscious actions. It's going to control smooth muscle, which you would find like in the walls of hollow organs, maybe like your bladder or within your GI tube, and cardiac muscle, as well as glandular secretions. Okay, the ANS is divided into 2 pieces. So we'll put a little number 1 next to the sympathetic and a little number 2 next to the parasympathetic. These are the 2 divisions of the autonomic nervous system. They have opposing effects. You can see here that the sympathetic is all about stimulation. So you can think about stress, when it comes to the sympathetic nervous system. Anytime you're stressed, then things are going to change within your body. Your heart rate may increase. You may have a burst of adrenaline, things of that nature. We'll get into a little bit more deeper when we get into the autonomic nervous system, but sympathetic is fight or flight. Okay? Ultimately, you'll hear that over and over and over. It's also freeze. So sometimes people leave off freeze. It's fight, flight, freeze. Okay? Fight, flight, freeze. Whereas parasympathetic is rest and digest. This is a relaxing effect. So it's not going to cause your heart rate to go up. It's going to cause your heart rate to go down. It's not going to cause your pupils to dilate. That's what's going to happen during a stress response. Your pupils will dilate so that you can see more clearly. Whereas in the parasympathetic state, whenever you rest, your pupils are going to start to constrict so you can go to sleep. So these are opposing effects in the autonomic nervous system. Most of your organs will have what's called dual innervation. That means most organs are going to have both sympathetic and parasympathetic input. Dual, D-U-A-L. Not all, most. Okay? Again, we'll talk about autonomic nervous system more whenever we get to that particular chapter. So this is just so you can see the different divisions as for right now. And then here's that enteric nervous system that you would find within the GI tract. 100 million neurons means that it has as many or even more than what's within the spinal cord. They're going to use the same neurotransmitters as the brain. We'll be talking about different types of neurotransmitters. Different ones cause different reactions within the nervous system. And they can initiate and coordinate these reflexes. Visceral, remember that means organs. They can coordinate these reflexes locally without need or input from the central nervous system. That means it can work on its own by itself, which is very cool because the nervous system pretty much controls everything. But the gut can do some of its own stuff without needing input from the brain or the spinal cord. It can be influenced by the autonomic nervous system, the ANS. And we see this whenever we look at GI diagnoses like irritable bowel syndrome. So again, we'll probably talk about that a little bit more as we get into the ANS. Okay, so that's your ENS. Let's talk a little bit more about ENS because it is so incredibly cool. There is this article right here. If you want to go and read the entire article, I just took snippets out, but it talks about how the gut is basically the second brain. So a lot of people don't realize how much your gut actually impacts your brain. And they don't realize how much food and mood are interrelated and interconnected. So I've said a lot about this throughout the semester already, but remember, when we're looking at our triangle of health, we have to have all three sides kind of come and work together. So this little brain inside of our gut can determine our mental state, and it does play key roles in certain diseases throughout the body. So it's very cool of how much this gut, this nervous system can actually do for your entire system, not just locally, but the whole entire organism. Okay? Depression treatments that target the mind can unintentionally impact the gut. This is very important when we're dealing with people in the psychology realm, because sometimes when they take medications, they feel like they're not getting better, they're actually getting worse. This is a really, really bad sign. So it's something that needs to be paid attention to, and somebody has to be able to step in and say, we need to look further and evaluate some things on a deeper level. So 95% of the body's serotonin is found in the gut. 95%. Serotonin is that happy hormone. Serotonin is often blamed for being in a very low quantity in people who have depression. However, this has not always been clinically proven to be true. So unfortunately, whenever people take SSRIs, these antidepressant medications, with the goal and the intention of increasing serotonin, sometimes it's really not that helpful because it causes these changes within the second brain that then cause a problem and an imbalance in serotonin levels. So the first brain is not helped. So we could look at these as going constantly back and forth, and some people will say, well, what came first, the chicken or the egg? Is the medication making it worse, or is the person themselves already getting worse? So again, that's why we want to take all parts of the triangle into consideration whenever we are dealing with patients, and not just merely say, well, you got this med, so you should be better, and if you're not better, then really it's just you. That's not fair to that patient. Okay, so you could see here that this goes on to also talk about autism and how many kids with autism also have GI impact. So I really like working with autistic kids because whenever you change the nutrition that they're putting in their body, it changes their brain, and it's amazing to see what can actually happen in those kids. There is this field called neurogastroenterology that if you're interested in things like this, like how the gut and the brain actually work together, I would encourage you to look more into this field, especially if you're not 100% sure what you want to do. Neurogastro is something that is relatively new. If I had the opportunity to go back to school, this is exactly what I would do. This would be the only reason I would ever go back to school, would be to be a neurogastroenterologist, because the amount of research that they do to see what's happening with the mind-body connection is just fascinating to me. So there's a lot more work that needs to be done. If you're interested in doing that, I highly encourage you to look up that profession and maybe dig a little bit deeper to see if that's where you would shine. Let's see, I want to get to, right here, 70% of our immune system is aimed at the gut to expel and kill foreign invaders. So you also have a lot of immunity that is important within your gut as well. So as we're talking about the nervous system, we don't talk about the ENS as much, which is why I wanted to throw this part in here, because the ENS actually comes more when we get into digestive system in 2402. But I wanted you to know how important this was and not forget about it, because we mainly focus on the CNS and the PNS and then the ANS. Okay, here's a little homework for you. Let me make this bigger so it's easier to see. Read this little article right here, and this is to help give you a better idea of that connection that I was talking about between food and mood, okay? Just a real quick 1 to 2 paragraph summary of your thoughts, what you learned from it, what you plan to do with that new information. Again, I hope that you are going to share it with other people so that way they can become aware of maybe it's not a medication that's needed, maybe it's a simple change or maybe an elimination of some things that they're putting into their body that would make them feel better. I will tell you, for me personally, this is one of the reasons why I cannot eat a lot of sugar, because if I eat a lot of sugar, then my brain goes a little bit on the crazy side, on what my husband calls the worrisome side. I noticed this whenever I, one day, for whatever reason, I don't even know why, ate just like a ton of sugar, and it was ridiculous what I was eating. I went to work and I thought that my kids were going to be run over by a bus. Like, it wasn't even time for buses to be out, but I was just anxious the whole entire time while I was at work. I thought my kids were going to die. I thought I needed to go home. I didn't know what was going to happen to them. And I'm like, what is happening inside of my brain? And then I went back and I thought about everything that I ate that day, and I was like, oh my gosh, that cannot be real. Did that just really just happen? So sometimes those anxiety, panic attacks, worrisome, is all related to what are you putting in your system, and then how is the system responding. So that's why I don't do that anymore, because that made me feel like, oh, I never want to feel like that ever again. So you have to be cautious. And alcohol also does this to people. So whenever they have the alcohol and they wake up in the morning and they think, well, this is just a hangover, and you feel like pure crap. Some people, it's not just a hangover, it's also a mental issue as well. So they have anxiety, nervousness, they have, you know, the heart palpitations, they start feeling really bad. Some of those things can be attributed to dehydration, but then other things would be attributed to the connection between alcohol consumption and what it does to your brain. So I hope you guys learn a lot about that food and mood connection, and maybe even identify some of your own triggers. So this is just a recap. So whenever we're looking at this chart here, what you can see is, again, we've already said the CNS is the brain and spinal cord in the gray. The PNS is everything outside of that CNS and the ENS. We have sensory information, sensory, afferent, SA. It's going to be the first step, right? Where do we get this sensory information from? Any of these places here. Special sensory receptors that are monitoring different senses that we learned in kindergarten. Visceral sensory receptors are going to monitor internal organs. Somatic sensory receptors are going to monitor things that are within our skeletal system and our skin. All of that information at any point in time can be delivered from the PNS into the CNS. So what I was telling you earlier, this is your step one. Okay, we have to have a detection of some sort of sensory information. This is step two. We have to integrate. What is it that I just received? What does this actually mean? And then step three, the motor is going to have to be sent out so that we can make a change based upon the information that was received. 1, 2, 3, every time. Okay? So within our somatic nervous system, again, you can see here, skeletal. Think skeletal. Autonomic, there's your two divisions, parasympathetic, sympathetic, smooth, cardiac, glands, adipose tissue. All right? Okay, so to briefly answer your question, this is not going to be like the full answer, but why adipose tissue? Because adipose tissue will also regulate hormones, especially estrogen. So whenever we're looking at the more adipose you have, in general, the more estrogen you're going to have. So for guys who generally have more testosterone, you know how they can lose weight relatively quick, that fat can fall off. It's not the same for females. So since hormones are all necessary for homeostasis as well, and sometimes that adipose is triggered because of its role with estrogen. Up and down with the adipose will be up and down with the estrogen as well. Okay, that goes a whole lot deeper, and we get into that more in 2402 with the endocrine system. Okay, you're welcome. All right, so now that we have that summation of all of those things, we're going to look at the neuron. We're going to look at the CEO. And as we're looking at this CEO, I'm going to have you guys draw a picture because I know for some of you, it's easier for you to learn with pictures. So I am going to try to see if I can pull up, yes, this whiteboard. And we're going to draw a neuron. I have many slides in here that will basically talk about the different parts of the neuron, but I want to draw it for you and I want you to draw it as well, so that way you can kind of remember some of these structures, and that way it'll be a little bit easier for you to answer questions based upon this neuron. So first thing that we're going to do is I'm going to start with the cell body. Okay? So draw something that looks like a circle. Okay, I think this is still screen sharing, so I think we're okay. And what you want to do is you want to label this the soma. Soma, S-O-M-A. That is the cell body for the neuron. On one side of the cell body, you're going to draw a straight line. This straight line, you want to label it as an axon. Okay, that is your axon. On the other side of your cell body, draw some squiggly lines, maybe something that looks like a little tree branch. Draw multiple, because as we are drawing, you're going to see that we're going to have more of these structures than we have of the axon. And that's very important. So these structures are called dendrites. Okay? Dendrites, soma, axon, and now we're going to draw different structure that kind of is at the end of the axon, and we're going to label this the telodendria. Telodendria. Now, maybe if you can use a different color, let me see if I can get a different color here. At the end of the telodendria, I want you to put a circle. My different color didn't show up. Okay, this circle at the end of the telodendria, you're going to label that as the synaptic terminal. That's specifically for that little circle. It won't let me change it to blue. Okay. Inside of the synaptic terminal, what you want to do is you want to draw tiny little dot. A couple of them. Tiny little dots. And these tiny little dots, you're going to label those as the neurotransmitters. Neurotransmitters. Since I'm at the end of my page here, I'm just going to label them NT.
Are they on both ends of the telodendria?
Are there what?
Are they on both ends of the telodendria or just on one side?
Oh, the
Synaptic terminal.
synaptic terminal is just at the very tip.
On both sides?
On both sides, yeah. Mm-hmm. Okay, so we have dendrites, a soma, a long axon. This is also sometimes called a nerve fiber. So if you're reading in your book and you read nerve fiber, you're like, what is that? That's the axon. The axon divides off and becomes telodendria. At the end of every telodendria, we will have a synaptic terminal. Inside of the synaptic terminal, we will have the neurotransmitters. I'm going to just draw an arrow so that you know this is the telodendria, and then we know that this is the synaptic terminal. And we know the neurotransmitters are inside the synaptic terminal. Okay? So this is our neuron. This is the cell body. What I want you to do now is I want you to underline the R in dendrites. Underline the R in dendrites. And right next to it, write the word receive. Receive. Okay? Then I want you to underline the A in axon. And right next to it, write the word away. And then draw an arrow, because what I am saying with this receive and away is when information is going to hit a neuron, it will be received at these dendrites, and then it will have to move this direction down the axon. So it comes into the cell body and then moves away from the cell body. This movement of this message is called an action potential, and we're going to get into that the next time, but the action potential will only go in one direction. So as I keep moving the arrow in this one way, one way, one way, one way, it's to reinforce to you that the information flow cannot go backwards. The information is going to be received here. It will move through the cell body along the axon towards the telodendria and then hit the neurotransmitters. And that is going to be the same direction every time. Okay? Now, whenever you're looking at this axon, what I want you to do is now we're going to add some boxes around just some portions of the axon. So it can't be one long box. It has to be multiple boxes. And these multiple boxes, you want to label those as myelin. Okay? The myelin is a protective sheath that surrounds the axon. One of the neuroglia is going to wrap the myelin around this axon. So we'll get into that in just a second, who's going to be doing that. And what I want to also draw your attention to is the space that is between each of the boxes. So in between each of the boxes, you have this space here that's called the node of Rambier. The node of Rambier is the space where there is no myelin. Okay? This all is going to become important when we talk about how the information is going to be transmitted along the neuron. We're going to get into that a little bit the next time, but for right now, I want you to be familiar with the structures, so that way you at least have that terminology down when we start talking about how information moves along the actual neuron. Anybody have any questions about this picture so far? Okay, so let's go back to our PowerPoint. So we looked at the cell body, right? We drew the soma. Inside of the cell body, it has all of the same stuff that is within every other cell that we've talked about in the past or that you've been through a biology class: mitochondria, ribosomes, cytoplasm, etc. So that's what's inside of the cell body. Whenever we're looking at dendrites, remember these are the ones that are going to receive information. These are those branched processes that come off of the cell body. All right, receive, receive, receive. The axon, we saw that. This is what's going to propagate those electrical signals, send the information on the neuron. Remember, that's called an action potential. We'll go through that next time. Inside of the axon, we have axoplasm, which is just basically the special word for the cytoplasm inside of the axon, and it has its own organelles in there as well. Here's a picture. So this is something like what your picture should kind of look like with your dendrites, your soma, your axon, the telodendria. What's at the tip of the telodendria? What do we call that?
Synaptic terminals.
Synaptic terminals. Good. So remember, terminal means endpoint. So that synaptic terminal is at the very end point of the telodendria. And then what lives inside of the synaptic terminal?
Neurotransmitters.
Neurotransmitters. Perfect. They are going to be the ones that are going to move the message from neuron to neuron to neuron. So when we're looking at this, it's very important that you know that as we look at the axon, in order to move that message, there's a couple of different terms that you want to become familiar with. The axolemma is the plasma membrane of the axon. So just as we have like a cell wall, you know, that surrounds each cell, this is a membrane that's going to surround the axon, the axolemma. The initial segment is at the base of the axon, and the axon helic is the region that attaches the initial segment to the actual cell body. Let me show you a picture so you can add it to your picture. So you can see here that axon helic is going to be where the soma actually starts to converge and it becomes a lot thinner so that we can lead into the initial segment of the axon. This becomes important in regards to action potential, which, like I said, we're going to talk about next time. So just make sure you add these two onto your picture, the axon helic and then the initial segment of the axon. Remember, the direction of the action potential is always going to be one way. And what you can see here is you see the telodendria, You see the synaptic terminals, but what I want you to point, or what I want to point out to you, what I want you to write down is that this is not actually touching this next cell in line, okay? So whenever you're looking at these synaptic terminals, even though it's drawn in the picture, it looks like it's touching this next cell in line, it's actually not. We'll get to that when we start talking about how information is transmitted across from one neuron to another. Okay? There can be collaterals along the axon, so there can be different branches. So like you can see on this one, they started to kind of draw a collateral. We drew ours as just one long nerve fiber, but it can have multiple branches coming off of the axon. Those are called collaterals. We have the telodendria, and we have the synaptic terminals, and AKA is axon terminals. You can use whichever term you want. I just like to use the term synaptic, so that way that you know that the next thing that's going to happen is the synapse, which we'll talk about later. All right, so, excuse me. The axonal transport, this is the movement of materials between the cell body and the axon terminals. So materials will move along the neurotubules within the axon, powered by mitochondria, and then a couple of other proteins that are going to help with the movement and the transfer of the material. When we're looking at different types of neurons, these are all CEOs, okay? All of these guys are CEOs. We have 4 different structural classifications. Which one of these did we draw on our picture?
The last one.
The last one, exactly. So what you drew on your picture, write down on your drawing, multipolar, okay? Your drawing represents a multipolar neuron. Over 90% of the neurons in the body are going to be multipolar, so I wanted to draw the most common one. Multipolar neurons, very common. These other 3, not so much. So we have an axonic neuron listed as A. What does A or an stand for in medical terminology?
Without.
Without, perfect, without. So you can see here it has no axon. Bipolar, that means we have 2 processes that are going to be separated by that soma. So you can see here and here, 2 separate processes. This is what makes this bipolar, bi meaning 2, polar opposites, okay? Unipolar, the soma is now off to the side, and so we have one long singular elongated process. And then of course the multipolar is the one that we drew. So these are the 4 structural classifications. We also have functional classifications. So these next few slides are everything that I already mentioned on the picture. If you want word slides, they're there for you. So our functional classifications are here. Sensory, AKA what?
Touch.
Afferent, right? And then we have motor, AKA what?
Afferent.
Efferent. And then we have the interneurons, which are going to be in between the 2. So our sensory neurons, we already said, afferent, these are the ones that will pick up information from the receptors. They're going to do the detection. A lot of these sensory neurons the afferent are going to be unipolar, okay? So you will see that commonly as a sensory neuron to be unipolar. Now, let me draw your attention to these words, somatic sensory. What do we say? Always remember when you're thinking somatic? Skeletal, perfect. So sensory information coming from skeletal areas, okay? From your skin, from your muscles, things of that nature. Visceral means that we're monitoring internal environments, so like the lungs, the heart, those things are going to be visceral. Got to keep those 2 separate because those 2 are going to be monitoring different parts of the body. We have different types of sensory receptors. So entero means that we're looking internally. Extero means what's going on in the outside world. Okay, it's not my blood pH and it's not my blood oxygen levels. No, it's the external environment, what's going on out there. What is there like a lot of smoke around me? Is there too much carbon dioxide? That's going to be an exteroreceptor. Proprioceptor means we're monitoring where that person is at in regards to space. Where is that joint at? As I walk back and forth in front of this computer, the reason why I don't run into a table or into the wall is because I know where my body is at in relation to space. So that's proprioception. Motor neurons, aka those efferent neurons, we're going to carry the instruction, remember again, from brain and spinal cord out to the peripheral effector so they can make a change, make an action. Number 3 on our list. We have somatic motor because we had somatic sensory, so therefore we must have somatic motor. We had visceral sensory, therefore we must have visceral motor. So whenever we're looking at, again, afferent, efferent, don't get those two confused because it will be so hard for you to remember what's doing what, where is it going, why is it doing that. Now we just added an extra layer on there with the somatic motor, visceral motor, somatic sensory, visceral sensory, okay? Okay, we'll talk about that when we get to autonomic nervous system, so we're gonna skip that for right now. Interneurons, most of these are gonna be in the brain and the spinal cord. Some will be found within the ANS, and these are responsible for, again, integration, part number 2. So this is where you're going to have your interneuron come in. It's going to say, okay, well, what was the sensory information that was received? What is the best action to take based upon that? So that interneuron is going to kind of be the intermediary between that sensory and that motor information. Okay. All right, now let's talk neuroglia. So you drew on your paper, what kind of neuron was it structurally? Multipolar, right? Over 90% of the neurons are multipolar. They are the CEOs. What was the neuroglia? Secretary, perfect. So now we're going to talk about the secretary for a second. Remember, their job is to support and protect those CEOs. So whatever they need, you need me to get your dry cleaning, you need me to get your coffee, are you hungry, you need me to go clean your office, whatever it is, those neuroglia are going to do. We have different types that are going to be within the central nervous system and the peripheral nervous system. So make sure you keep these two things separate. These are the 4 in the CNS, okay? Brain and spinal cord have these 4. You will not find these 4 in the PNS. If you like charts, there is a chart right here that will tell you in general kind of what their functions are, the 4 within the CNS. I'm going to go through the word slides because I feel like I always go through charts and pictures. So for word people, I'll go through the word slides. So the astrocytes, very important because these are going to help maintain that blood-brain barrier. And we'll talk about the blood-brain barrier a little bit more when we get into brain, but we want that to be very tightly regulated so that we don't have any external factors coming into the brain that could cause injury, infection, or any type of issues. So the astrocytes have a very large role in helping to maintain the integrity and the strength of this blood-brain barrier. They're also going to be responsible for repairing damaged nervous tissue. So that's very interesting. They will form scar tissue. That's the unfortunate part, is that whenever they start doing that repair, it just becomes scar tissue. Remember what I told you guys in the very beginning, if we change the structure of something, then we're going to change the function. So This is where we can kind of get into some of problems within the neuron repair and regeneration within the CNS. It's very rare to have that repair and regeneration within the CNS successful. It's more possible in the PNS.
So if you lose some of those, then you're pretty much like they're just gone.
Yeah, so if you lose them, then that's it, they're gone. You can train different parts, like let's say brain. Say you have a stroke, and those nerves are gone in one particular area that doesn't allow you to speak. You can retrain a different part of your brain, like through singing. So sometimes for stroke victims, they'll train them to sing if they have the part of their brain that no longer allows them to produce speech. So they'll use different ones to kind of cover up for the loss of, but it never really completely comes back. Yeah, unfortunately. So here's a picture of an astrocyte, and what I want you to just see here on this particular picture are these little foot processes. See these little foot processes? They're just kind of like anchored on here. This is going to help forming a very tight and regulated ability that they have. So like if you see it right here on this blood capillary, you see how they are just all lined up with one another. So this is why there can be such tight regulation of that blood-brain barrier, because their little foot processes all connect to one another, and they just basically do not allow very specific material to seep into that BBB. All right, the epinemal cells, the important thing you want to remember from epinemal cells is that these are going to be highly, highly important when it comes to our CSF, our cerebrospinal fluid. So the epinemal cells will line what's called the central canal of the spinal cord. I know we haven't learned that yet, but you will. And the ventricles of the brain. You'll learn that as well. They're going to line these areas, and it's within these areas that we get this fluid that's called the cerebrospinal fluid. So they're going to help secrete this fluid and ensure that we have enough of that CSF on a regular basis. We'll talk more about the function of CSF whenever we get into the brain. It's absolutely amazing what this fluid does. So here's our little epinemal cells that are just these little simple cuboidal epithelial cells that just kind of line up and help create this CSF. Oligodendrocytes, these are going to be important for you to remember that they create that myelin sheath that is going to surround the axon. So whenever we're looking at the picture that we drew for that multipolar neuron, because that's going to be a motor neuron that would be out in the PNS. So it's not the oligodendrocyte that would be doing what we drew on a picture. I'll tell you which one it's going to be here in just a second. But the oligodendrocyte will do this within the CNS. Okay, myelin not only helps protect the axon, it helps increase the speed of information transmission. That is one of the most important parts of myelin. So put a big star right there, increases speed of action potentials. That is one of the benefits of having a myelinated axon. Not every axon in the body will be myelinated, but those that are going to be able to have a lot faster information transmission. Okay, oligodendrocytes, because they do the myelin, then we have the nodes, which we kind of drew on our picture, the nodes of Rambier. White matter, when we're looking at white matter, regions of the CNS with many myelinated axons are going to be referred to as white because the myelin makes it look white. So to keep that in your brain will be important as we go through the brain and the spinal cord, because you're going to hear this over and over and over. White matter, gray matter, white matter, gray matter, white matter, gray matter. So come back to this slide if you forget. White matter, many myelinated axons. Gray matter, it's either going to be unmyelinated axons or the soma or the dendrites is what we would be looking at, or what would be referencing if we're saying that something is gray matter. Okay? So there's a picture of an oligodendrocyte. You can see it all the way over here. You can see it's just kind of surrounding and wrapping around that axon in order to protect it, but not completely. Remember, that's why we get the nodes. All right, and then microglia, the fourth one, smallest and least numerous. This is going to be like the MAID. So remember, microglia starts with an M, MAID starts with an M, because this is the cleanup guy. So in the body, we have these cells that are called macrophages. And macrophages do a wonderful job of helping to clean up things, but they're not going to be within that central nervous system. So the microglia are going to be what are going to be cleaning up the cellular debris, waste, pathogens, etc. Okay, so there's a picture of your microglia with its vacuum cleaner, its sponge, its 409. Do you guys see it? No, I'm just kidding. All right, so that's your MAID, your cleanup MAID. Now, neuroglia of the PNS, only 2 types. So you have 4 in the CNS, 2 in the PNS. The 2 in the PNS are called the satellite cells and the Schwann cells. I have another little chart for that. So if charts are your thing, then there you go. Words are your thing, then here you go. Satellite cells. Sometimes these are compared to like the astrocytes within the CNS. It's been, you know, more work that's had to be done in regards to satellite cells, like what is their actual function for, because we didn't know for a while. So right now, just remember, regulate the interstitial fluid around the neurons, keep things healthy and well. Schwann cells, super duper important, put a star next to these guys, because Schwann cells are the ones that you actually drew on your picture to create that myelin sheath around that multipolar neuron. So when we're looking at Schwann cells, it says right here, a myelinating Schwann cell sheaths only 1 axon. That's because oligodendrocytes, they can do more than one. They can kind of stretch out and they can do like 1 on the left and 1 on the right, but a Schwann cell can only do 1. So it doesn't get to do that double duty. And the way that it does its work, so again, here's our picture of kind of like what we drew. This might be something you see in lab, but let's get to this one. The way that it does its work is it just wraps around that axon multiple times over and over and over again. So think about this blue axon as like that centerpiece of cardboard in your wrapping paper roll. Whenever you're wrapping presents for Christmas, this is that centerpiece of cardboard, and you can see that it just wraps around multiple times. And this is how it actually will insulate and protect that axon, is that there's so many rotations that's wound around this axon in order to provide that protective benefit to it. Okay, so that's your Schwann cell. Now, if we have an injury, remember in CNS it's really not going to regenerate, but in the PNS it does have opportunity to. But if it does not, then Walarian degeneration is what will happen if the axon distal to the injury starts to degenerate. So what does that mean? Whenever my husband had his motorcycle accident, his bone came through his ankle. Basically his fibula. His fibula came through his skin, his foot turned around completely backwards. So that means muscles, tendons, ligaments, nerves, all torn in that area. So he no longer has the ability to pick up any sensation from that part of his leg where that major injury occurred. And this is the reason why. It's because whenever you have an injury, anything distal from that shearing force or that injury site, anything beyond that is going to degenerate and you will no longer have function there anymore. So this is like if you start to see it fall apart right here, this is step 1. What's going to happen is the Schwann cells are going to try to put it back together. That's going to ultimately be the goal. See, the macrophages, the cleanup crew is going to kind of come in and kind of clean up some of that debris because there's going to be a lot of things that are going to need to be cleaned up whenever you have that type of injury. So as the Schwann cells try to put this thing back together, what you're going to see is ultimately we end up with something that looks like this. Okay? If you change the structure, you will change the function. Okay? So he can't feel there anymore because it's not coming back. And we don't expect it to ever come back. It's been years now. And it's only going to probably get worse for him as he goes through more surgeries to continue to take care of it. So whenever we look at the degeneration, just remember, ultimately, this is what's going to happen whenever you have that type of crush injury, shearing injury, fracture, bone break, something like that. Okay? Now, before we go into the next class, I want you guys to go look at this crash course. I know not everybody likes that man's voice, but you want to watch something in regards to action potentials before you come into class because it is so heavily loaded with multiple different, what's the word I'm looking for? Multiple different, it's like chemistry, it's physics, it's engineering. What's the word?
Multi-disciplines.
Disciplines, thank you, that's the perfect word. It's multiple different disciplines, and if any of those disciplines are not your strong suit, it makes it very difficult to understand because you have to know all of the disciplines at the same time in order to understand action potential. So do watch this crash course or something else on action potential before we come in, so that way you can kind of already be a little bit familiar with it. And I hope to be able to maybe break it down for you and make it a little bit easier so that you can understand it at a better level.